Dr. Mukhtar, David, and classmates. This is my bonus genetics video on DNA replication, transcription, and translation. So first, let's ask ourselves, what is DNA? Deoxyribonucleic acid is a molecule made up of deoxyribose sugar, phosphate groups, and nitrogenous bases that contains the genetic information in most living organisms. DNA must replicate, transcribe, and translate in order to sustain the genetic information needed for life. First is replication, which essentially is DNA doubling before cell division. Replication occurs in 4 simple steps: initiation, unwinding, elongation, and termination. Initiation. In order for replication to occur, we must make our double-stranded DNA into single-stranded templates. First, initiator protein binds to the replication origin. A short piece of DNA then unwinds. DNA helicase then comes in and breaks the remaining hydrogen bonds between the 2 nucleotide strands, which runs from 5' to 3'. Single-stranded binding proteins then attach to the single strands to prevent secondary structures from forming. DNA gyrase is another essential enzyme that helps relieve torque built up in the unwinding DNA. Next is elongation. The enzyme primase adds an RNA primer because a 3' OH group has to be present in order to initiate DNA synthesis. A primer is added before every Okazaki fragment begins. An Okazaki fragment is the gap between continuous and discontinuous DNA, or the leading and lagging strand. DNA polymerase III then adds new nucleotides to the growing 3' end. DNA polymerase I is also an essential enzyme that removes RNA primers and replaces them with DNA. After DNA polymerase I has replaced a primer, DNA ligase is then used to seal up the gaps in the new DNA strand. The top is a picture with the essential enzymes in the leading and lagging strand, and then this video is a computer imaging model to show you what replication looks like. Last is termination. It occurs in 2 ways. Either 2 replication forks can meet, or a termination sequence blocks helicase from moving down a DNA strand. So now what's after replication? Transcription. Transcription is the synthesis of an RNA molecule from DNA. A transcription unit must be present, which includes the following: a promoter, RNA coding region, and a terminator. The promoter recognizes template DNA and is the start site. RNA coding region is a sequence of DNA being copied into RNA, and the terminator is the end site. Similar to replication, transcription occurs in 3 steps. There's initiation, elongation, and termination. The transcription apparatus, which consists of several important enzymes, binds to the promoter region. RNA polymerase is then able to bind to the promoter to begin synthesizing RNA. RNA synthesis occurs from the 3' end of an RNA molecule and is complementary and anti-parallel to the template DNA strand. Elongation occurs when RNA polymerase continues to unwind DNA as nucleotides join to the growing RNA strand. Termination. Once RNA polymerase transcribes the terminator, transcription stops. RNA polymerase detaches from the DNA template, then RNA detaches from the RNA polymerase. So, next step is translation. Translation is the process of assembling amino acids into proteins, which essentially is protein synthesis. Translation occurs in 4 steps: tRNA charging, initiation, elongation, and termination. tRNA binds to a specific amino acid on its 3' end. Recognition of the appropriate amino acid is based on size, charge, and R group of the amino acid. mRNA binds to the small subunit of a ribosome. Initiator tRNA binds to the mRNA by base pairing between the codon and anticodon. The large subunit of a ribosome then binds and forms the initiation complex. Elongation. A ribosome has 3 sites that tRNA can occupy. E site, which is exit, P, which is the peptidyl, and A, the amino acyl sites. The initiator tRNA starts out occupying the P site. Another charged tRNA comes and binds to the A site. The amino acids on each of the tRNAs form a peptide bond, which allows the amino acid to be released from the tRNA in the P site. The last part of elongation is translocation. The ribosome moves down the mRNA strand to the next codon. The tRNAs in the existing P and A sites then shift to the E and P sites to allow another tRNA to occupy the A site. The new amino acid forms a peptide bond to the other existing amino acids, resulting in a growing chain. Lastly is termination. Elongation continues until a termination codon is translated. Once this codon is translated, a release factor binds to the A site in the ribosome. The release factor causes the tRNA and the mRNA to be released from the ribosome. Here is a recap of translation in general. Thank you for watching, and I hope you guys enjoyed.